So I wanted to tell you about topological defects in active and living matters. Topological defects, as I'm sure you know, play a central role in the physics of many materials, from magnets to superconductors to liquid crystals. And what I would like to show you today is that they really acquire new life in active matter. In active fluids, they become motile and they drive essentially the turbulent-like motions that one sees in a lot of active systems. And in active solids, they actually are correlated with shape changes and morphogenetic processes. And Lucilla thought this was a cactus. It's not a cactus, but is our rendering of actually an organism called Hydra. And I will tell you about that a little later. It's a great model system for studying the role of defects in active nematic solids. So, well, active matter is essentially matter composed of entities that consume energy to self-organize in a variety of emergent structure on a broad range of scales. I'm sure everybody in Italy is very familiar with flocking, thanks to the great work on this done in Rome, and that's an example of an active phenomenon. But other examples may be the collective migrations of cells in epithelial monolayers, or even inside a cell, the organization of protein and actin fibers to drive, say, cell deformations and cell motility. One feature that is common to many active systems is that the active particles, that we could call them, are actually elongated, and so they can organize in states with orientational order. And generally, these systems exhibit a variety of type of self-sustained dynamics, or they can drive spontaneous shape changes. So in this talk, I want to focus on a particular type of order, which is nematic orientational order. A passive nematic liquid crystal is essentially a fluid of rod-like molecules that can form a state where the molecules on average have a random orientation. That's an isotropic liquid. Or as you either lower the temperature or increase the density, can order in a state which has a nematic order. That is, these molecules are on average aligned in a common direction. And this type of order is characterized by generally a unit vector called the director. But due to the fact that the rods are heterosymmetric, the order state has inversion symmetry, that is a symmetry for n going to minus n. And so the order parameter, unlike in ferromagnets, where the order parameter is a vector, here the order parameter is a rank 2 tensor that we call the Q tensor, characterized by a scalar magnitude of order, and this director field. Now, active nematic liquid crystals are fluids composed of elongated agents that exert forces on their surroundings, and as a result, maintain these self-sustained flows. This example here is a suspension of bacteria, Myxococcus xanthus, from the lab of Josh Shavitz at Princeton. And the color denote regions that are oriented in the same directions. The forces that these agents exert, this bacteria actually kind of goes back and forth. That's why it's nematic and not polar. And so they exert force dipoles. And so there is no net force, but there is an average stress exerted by these agents on the system. And that's what we call active stress, and it's proportional to the orientation order parameter through a prefactor that I will call activity, that is controlled essentially by the biomolecular processes that drive the motion. For instance, in this case, the concentration of bacteria, the speed at which they move back and forth. The sign of this alpha controls whether the forces are extensile, that is, the dipole pushes out, as in this example here, or contractile, that when alpha is positive, as for instance in actomyosin gels. So that's an active nematic liquid crystal. And the reason I want to focus on nematic order is that nematic order has actually been seen quite ubiquitously in active and living matter. The sort of paradigmatic example is this one to the left, which is a thin film suspension of microtubules, long, stiff filamentary protein extracted from living cells, cross-linked by clusters of other protein that are able of use, transform chemical energy mechanical work, and essentially move these filaments relative to each other, driving these turbulent-like flows that you see here. Similar type of dynamics is observed in both dead and other living systems, but even in dead systems. This one here is I hope you can see my cursor. And also, by the way, please feel free to interrupt me with any question. So this is a monolayer of a granular rod vibrated vertically. This is a dense layer of epithelial cells that flow spontaneously in a channel. And you see nematic order even at the scale of entire organism in this freshwater polyp hydra that I will tell you more, as I promised, about later. So there's a lot of nematic order in active matter, but of course, like in passive systems, order is never perfect. If you drive a system through the transition, say, from the azotropic to the nematic state, generally there might be order regions interrupted by topological defects. And the topological defects are true fingerprints of the broken symmetry in the sense that the lowest order topological defects depend, for instance, on whether the order is polar or ferromagnetic, in which you have vortex, anti-vortices of integer charge, or nematic, in which the lowest order defects actually are so-called disclinations, have this characteristic structure here, and they have a fractional charge. The charge is defined as the ratio of the angle through which the order parameter winds when you circle the core of the defect by 360 degrees. So these comet-shaped-like defects has charge plus 1F, and these trifold symmetric defect has charge minus 1F. Now, in equilibrium, one can show that the elastic distortion associated with the presence of the defects leads to an interaction among defects, which is really like a Coulomb interaction, and that's why the name topological charge. And so in 2 dimensions, most of my talk will be focused on 2 dimensions. Opposite sign charges attract with an interaction that is logarithmic. And eventually, a long time, they will annihilate and the system becomes ordered. In active systems, however, situation is quite different. First of all, the state of so-called active turbulence, which, by the way, is actually really be called spatio-temporal chaos, because it occurs essentially in a regime of 0 Reynolds number. In this state, one actually has a steady state number of topological defects as opposite sign pairs are continuously created and annihilated. And furthermore, as you can see here, the defects become motile. This is particularly evident in this set of still images to the right, where the arrows point, the blue points to the minus 1F and the red points to the plus 1F. As time goes on, you see that instead of being attracted to each other through Coulomb interaction, they actually move away from each other and they do not annihilate. So we have a steady state number of topological defects that essentially generate the distortion that drive and sustain these flows. And opposite signs pairs seemingly repel. And this type of motion has been seen in a number of systems, like the 2 included here. Now, in biology, there have been a number of experiments showing that correlations between the location of topological defects in bacterial layer or in dense layer of cells and specific biological functions. For instance, in bacteria, it has been demonstrated that the location, if you have a single layer of bacteria and there is nematic order, you can identify the location of the plus 1F defects and show that that's the place where the bacteria, as they keep dividing, start forming a second layer. These are, this bottom here is again Myxococcus xanthus by the same group of Joshevitz at Princeton. In layer in tissue, in epithelial monolayer, the location of the +1F has been correlated with the place where the cells die and get extruded from the monolayer. In this polyp hydra, the group of Kinneret Keren at Technion in Israel has actually shown a correlation between the location of topological defects and the specific morphogenetic processes, such as the formation of these tentacles or the formation of the foot, the formation of the mouth. And this is another example of a constrained cells that actually form a finger-like protrusion at the location of a +1F defect. So there's a lot of interest in trying to understand what drives this kind of shape changes and phenomena in general. So with that kind of longish introduction, what I would like to do today is tell you about topological defects, first in pneumatic fluids, how defects can become motile in active pneumatic fluids, and drive these turbulent dynamics and how we might actually control them. And then, hopefully I have time, I will tell you about they can actually become motile also in active pneumatic solid and the role they might play in the development of these polyp hydra. The work I will mention has been done over several years with a number of collaborators, but in particular, a former student, Suraj Shankar, now on the faculty in Michigan. If you haven't had him as a young speaker, I highly recommend it. A former undergraduate, now a graduate student at the University of Chicago, Luca Scharrer, and there is a C missing in his name. My collaborator, Mark Bowick, a former postdoc, Fridtjof Browns, now at KITP, and a current student, actually, Zhiheng Wang. So let's talk about defects in active pneumatic fluids, first of all. How we go about modeling an active pneumatic fluid, first of all. We have one way of doing it is at the continuum level, writing down essentially hydrodynamic equations in terms of the order parameter Q and the flow velocity that I will call U. So the order parameter essentially has dynamics that is controlled by relaxation, controlled by sort of a typical double well potential that controls whether you are in the isotropic or pneumatic state. There is an energy cost for deformation of order. So this elasticity constant K, I'm assuming just one elastic constant for simplicity. So in reality, the system is anisotropic and there is more than one. So this would be the derivative of the traditional Landau-Dejean free energy used to describe passive pneumatic liquid crystals. In addition, clearly flow has an important effect on the orientational order. Flow can, of course, advect the order parameter, but in addition, shear flows and vorticity omega can clearly distort order. And so that's characterized in these terms over here. The flow velocity, you could write the Navier-Stokes equation, but I'm interested in films which are generally on a substrate and really are in a regime where inertia is not important. So I really just write a Stokes equation of force balance. So I balance friction with the substrate, with pressure gradients, viscous dissipation, and elastic and active stresses. Elastic stresses, again, are associated with this elasticity K of the liquid crystal. And the active stresses come from these force dipoles that I mentioned earlier. So the key point of these equations is that there is really a feedback between flow and orientation. Active stresses, which come from distortion of the order, such as defects, act as a source for flow, and flow distort orientation, and the feedback between the two leads these self-sustained dynamics, essentially. And if you take these equations and you simulate them, you solve them numerically, you find a behavior which is very similar to the one observed in experiments. These two colorful images describe both the flow and the orientational order. The one to the left, in the one to the left, the color is the degree of order, The blue is 0 order and red is high order. And defects, of course, the defect cores, it's where the order goes to 0. And so you see the defect pair being generated in this blue region. To the right, the color is vorticity. And so you see these large-scale vertical flows with defects getting unbound in the high shear region between counter-rotating vortices. This type of dynamics has been studied over the years by many, many people and characterized in many ways. But one important piece of characterization I would like to point out is that there is a characteristic length scale that we call the active length that comes from balances active stresses with elastic stresses. So it goes like the square root of the nematic stiffness to the strength of activity. And this characteristic length scale controls both the defects and the active flows in the sense that the defect spacing is characterized by these length scales, and so is the size of the flow vortices. And this has been verified in experiments, this kind of behavior with the inverse square root of activity of this length scale by measuring both the velocity correlations, the separation between defects, and the correlation of vorticity. So this suggests really I would almost call a duality, in quotation, between flows and topological defects that suggests that one may be able to really describe these active flows by looking at the dynamics of the defects. And in fact, it's rather easy to understand why defects become motile in active system. I've written here a very simplified version of the Stokes equation, controlling force balance, that displays perhaps more clearly that active stresses act as a source for flow. So if you insert on the right-hand side of this equation the texture, the form of Q corresponding to, say, a -1F or a +1F topological defect, you can then solve this equation and find the flow that such a texture generates. This is just a linear equation. You can actually solve it analytically. And what you will find is that the flows are displayed here. Again, color is vorticity, and the black lines are the flow line, the streamlines of the flow. So what you see is that the +1F defects, which has this polar comet-like structure, generates a flow consisting of these two counter-rotating vortices. But the important point is that the flow the defect generates is finite at the core of the defect. As a result, the defect really rides along kind of like a colloidal particle with the flow it itself generates. And the velocity at the core is controlled by the size of activity. And the direction is actually controlled by whether the active forces are extensile or contractile. So the +1F defect really behaves itself as a self-propelled polar particle. If the forces are extensile, is expected to move towards its head, and for the forces are contractile, is expected to move towards its tail. The -1F defects generates an even more complex flow with actually 6 vortices, but the flow vanishes by symmetry at the core. So to leading order, this is a passive particle. And one important thing here is that the sign of activity alone determines the direction of the motion of the +1F defect. This has actually been quite useful, especially in biological setting, to try to understand the nature of the forces that drive active motions, which can be in biology either contractile or extensile, depending on the system you use. So here, what I'm really doing is thinking of defects as sort of quasi-particles, as well established in equilibrium, where we use a description of defects as Coulomb charges to understand order-disorder phase transitions in 2 dimensions, as essentially coming from defect unbinding. In fact, you can go further, and this is actually the work that Suraj did a few years ago. You can really, so as I mentioned, in equilibrium, you can actually map the defects onto a Coulomb gas, and that's how you formulate the theory of order-disorder transition, known as the Berezinski-Kosterlitz-Towles transition. What you can do in these active systems, you can actually map the defect dynamics onto a gas of particles that again interact like Coulomb charges, but the plus 1F are self-propelled particles and the minus 1F are actually passive particles. So in addition to worry about the dynamics of the core of the defects, you actually have to worry about the torques that the self-propelled particles, the plus 1F that are the comet-like polar defects, exert on each other. So we actually derived these equations, even though I wasn't planning to show them to you. And we actually showed some years ago that this transition to this state of spatiotemporal turbulence, or what we call sometimes defect chaos, in the plane of activity and nematic elasticity can actually be understood as a activity-driven sort of BKT type of unbinding with, again, this V naught will be the propulsive speed of the plus 1F defects, which is controlled by activity, and it renormalizes the effective stiffness proportional to the square of activity, in a way proportional to the square of activity, and this parameter dr controls the noise in the system that makes essentially the noise in the system. And these non-equilibrium BKT transitions has some interesting feature that are common to BKT transition, but there is no universal jump in stiffness, although there is still an essential singularity of the correlation length, and there are some interesting analogies to work done on driven dissipative Bose-Einstein condensates here. And this is rather technical stuff, so I just wanted to briefly mention, but I will like to, if you're interested, I particularly recommend the second paper back in 2019. Nowadays, so this is work from some years ago, nowadays there is actually a lot of interest in trying, due to this duality between essentially defects and flaws, there is a lot of interest in trying to control actually defects in order to tune and design active flows, essentially try to rectify the flow instead of having turbulent flow, having flow that you can actually design. And experimentally, this actually can be done by using, in these microtubule suspensions, for instance, by using motor protein that become active only when you shine light on them. And this video here shows work done by at Caltech, where essentially they have generated a +1 topological defects composed of microtubules. And you see this little yellow patch is the light, so the activity is controlled. The system is only active when there is light, which is this yellow patch. And by moving this yellow patch, they're able to actually control the location of the defect. And we have done something similar in theoretically, if you like, in numerical simulations. We make patterns of activity that essentially are able to grab the topological defects. We call them topological tweezers. And we are able to move the defect around as we like. In this case, we are actually braiding the image to the right is the path of the defect. So we're actually braiding the defects. And the goal of this is, as I said, to try to figure out patterns of activity that will allow you to design the flows. This is very much ongoing work, and I just wanted to kind of mention it as an interesting direction in which some of this work is going. But so that's about active defects in active pneumatic fluid. It's a very kind of quick review of what are the interesting things there. Main thing being these defects become motile. They can be described as quasi-particles, and there is a close connection between defect dynamics and flows. Now, what The rest of the talk, I would like to talk about more recent work on active nematic solids, which was mostly motivated by this work done by Kinneret Keren at Technion on hydra morphogenesis, hydra development. And in particular, tell you how the mechanism through which we believe defects can become motile in solids as well, and then control morphogenesis. But before I do that, maybe I'll just stop a minute, see if there are any questions on what I've been discussing so far.
Are there any questions?
Okay.
I don't see any hand raised, so you can go on.
Okay, let's talk about hydra. What is hydra for a minute? So hydra is a freshwater polyp. You see kind of a little image of hydra over there. It has a body, it has some tentacles, it has a foot, it attaches with a foot to substrates at the bottom of the creek or something like that. And one of the remarkable properties is that you can cut off a little piece, a little fragment of hydra from the side of the body, and it will regenerate within 2-3 days. It will regenerate the entire organism. This video here shows how a little fragment has been cut off, and you see that it kind of twitches. So the fragment first falls onto itself. It forms like a spheroid, and the muscle fibers on the surface of the actin body, first of all, are quite disordered. Then eventually they order, the spheroids elongates. And as I said, in a couple of days, the timestamp here is actually hours. In a couple of days, after quite some twitching, it kind of regenerates the entire organism. As it does so the body of hydra, this is just shown schematically, but I will show you an image in a minute again. It's actually composed of a bilayer of tissue. And on the outside, you have these fibers, which are really fibers of actin that span many cells. They connect together and span many cells and acquire really a high degree of nematic order. And experimentalists have noted that the correlation between, at the beginning, the system is disordered. There are a lot of plus and minus 1F defects, possibly even some plus 1 defects. Eventually, a lot of them annihilate. Because you're on a sphere, you cannot have perfect nematic order, so you must have at least a total charge of plus 2, which can be accommodated, for instance, by 2 plus 1 defects at the 2 poles. And they have established the correlation between the location of the defects and the location of the formation of, say, the foot, the head, and the tentacles. And so the idea is try to understand how this regeneration occurs and what is the role of nematic order in the process. Now, it's also well established that it's not all mechanics. There is a morphogen, a chemical called head morphogen, which is expressed at the location of the future head, and that plays clearly a role in the selection of the body axis, where this thing goes from being just a spheroid to being an elongated object, then it selects a body axis. This little bud on the side has to do with how hydra actually reproduces. It makes little bud on the side, and then this will become the head of a baby hydra, if you like. So in addition to mechanics, we actually have to worry about the role of chemistry, the role of this morphogen. But essentially, once the organism is fully formed, as I said, this Kinneret Keren and her group have established a correlation between the defects. And this nematic structure that you see at the surface is characterized by essentially what you have is a plus 1 defect at the head. Next to each tentacle, you have 2 minus 1F at the base of the tentacle and the plus 1 defect at the tip of the tentacle. You have 2 plus 1F very close to each other, or almost in the formation of another plus 1 at the location of the foot. And you add up all these defects. Of course, in the mature hydra, they have to add up to plus 2, which is the topological charge required to accommodate nematic order, that is lines, on the surface of a sphere. So the point here that defects are clearly tightly correlated with the body plan, the formation of the various structures, and we have to worry about the role of this head morphogen. Another important observation is that as the hydra is regenerating, there is essentially no cell division nor cell intercalations. That is, the structure of the actin fibers, which you could think of as muscle fibers, the structure changes, but cells do not change neighbors. They don't change location relative to each other. And this is kind of demonstrated, for instance, in this video, where what is highlighted in these little squares is the location of 2+1F defects. And if you look some hours later, this is 13 hours, this is 50 hours, the defects have become a single +1 defects, but the cells are really highlighted in green have not moved. So in other words, we have a restructure of the texture without cell motion. So this is not a fluid, but is more like an active solid. And in order to try to model this kind of active solid, we really have to worry about the interplay of mechanics, geometry, the expression of this morphogen, and how they feed back onto each other to regulate the alignment of these actin fibers during development. So what I would like to do for today is try to address two first simple questions. First, I will look at a simple model that addresses the question of how geometry and chemical activation due to the morphogen interplay in regulating the nematic texture. So I'm doing one leg of this loop. And I will then try to show you how what is a possible mechanism for defect model motion and reorganization in this active solid that can happen actually without any flow of material. So quite different from what happens in the fluid. So let's start with the first question. And for this, this was work done by Fridtjof Braun and Ji-Heng Wang. And for this, I will start with really the simplest possible model. In fact, this is for now a completely equilibrium model. And so I write a free energy in terms of the nematic order parameter and the concentration C of the morphogen. So this first term is the elasticity of the nematic, essentially says that the actin fibers tend to align with their neighbors, tend to suppress gradients of Q with some energy cost K, is my elasticity that I introduced before. The second term in green embodies the tendency for the nematic to align with morphogen gradients. This has actually been established experimentally by doing interesting experiment where they graft together pieces of hydra with different orientation. They regulate morphogen gradients, and they see essentially this effect that the nematic structure tends to align with morphogen gradients. Then I say that the dynamics simply also occurs through relaxation. So I actually look for states that minimize the free energy of the system. Actually, for at very first, let me even forget the morphogen and just ask how energy gets minimized onto, say, a spherical or an ellipsoidal surface. So let me ask about the role of geometry and shape, first of all. I already mentioned before that, but make it clearer now, that topology, that is a spherical structure, requires a topological charge of +2. And this could be accommodated by either having 2+1 defects at the two poles of the spheres, or actually having +1F defects, which will be located at the corner of a tetrahedron inscribed by a sphere, sort of like in the structure of a baseball. In equilibrium, the 4+1F structure is actually energetically favored due to the Coulomb repulsion among the defects. But if you and that's the structure I was showing here on the sphere. If you actually try to stretch out the sphere into an ellipsoid, you find that the geometry forces, drives the +1F defects to be closer to the pole, pushes them closer to each other, that this geometry can kind of overcome the effect of Coulomb repulsion. And you can, again, simply look at geometry sort of by hand, put in the geometry of Hydra, which Lucilla called a cactus. It's not a cactus, it's our attempt to draw the geometry of Hydra with a couple of tentacles over there. And what you will find is that this actually, you do get a defect structure similar to the one found in Hydra, but with one important omission, which is that we do not get any defects at the location of the future head, where experimentally one observes a +1F defect. So geometry and nematic elasticity alone are not enough to reproduce the texture observed in Hydra. So now we're going to put back our morphogen gradient and see what the role of the morphogen gradient is. Now, for simplicity, let's actually go back, first of all, to the plane. So now I'm going to look at the interplay of elasticity and morphogen gradient, but I'm just going to do it in the plane. Now, let me start for, so what I'm going to do is assume an order structure and prescribe a morphogen gradient in this region, inside this little circle, the orange circle over here. I'm going to prescribe a morphogen gradient which has a Gaussian profile, which some radius are not. And what this image here shows is the magnitude of the morphogen gradient, and with the circle being where the gradient is largest. So the morphogen gradient, remember that the Q tries to align with the morphogen gradient. So as I ramp up this parameter beta, that is the strength of this coupling, that's what I'm doing here, the nematic gets distorted. And eventually, the morphogen gradient leads to unbinding of two plus-minus defect pair. I mean the plane, so the net topological charge has to be 0. And that's why I need to have both plus and minus pair. So this to the unbinding of the plus and minus pair, but also allows the you get this configuration where the two +1F defects are actually end up being quite close to each other. That is, the morphogen gradient kind of overcomes a little bit of the Coulomb repulsion between the +1F defects. So this was in a plane where but it's suggestive that something like this may happen at the head of Hydra, where this morphogen is really expressed. And so, in fact, now I use the same free energy, but I change the geometry and topology, and I put now my nematic texture on a sphere. And you're going to see two views of the sphere, the top of the sphere and the bottom of the sphere. And the top is where I will impose a morphogen concentration profile, again, a Gaussian like I did in the plane. And so what you should see here, okay, now the morphogen is blue here, sorry, instead of being green. Okay, this was a bit too fast. Let me see if I can go back. Okay, so you see that the two defects kind of, the two +1F defects become very close to each other, acquiring almost the structure of a +1F defect, while in the bottom of the sphere, they remain relatively well separated. If I, however, also elongate the sphere into an ellipsoid, then at the top where the morphogen is being expressed, the tendency to align with the morphogen allows together with the geometry, allows the system to overcome Coulomb repulsion, and you get a +1 defect. And at the bottom, the two +1F, where there is no morphogen, but the two +1F defects get pushed pretty close to each other. And if you actually solve this energy minimization problem on the same surface structure that mimics the behavior of Hydra, you actually are able to reproduce the texture that is observed in Hydra. And for kind of fun, we also put a little spot of morphogen here on the side of Hydra, where it leads to the generation of these structure with a +1 in the middle and to -1F on the side. And that's where a bud will come out, possibly leading to a new organism. It's really the same mechanism that actually leads to the generation of the tentacles on the side. So the idea here is that by putting together simply elasticity of the liquid crystal, morphogen gradients, and geometry, we're actually able to reproduce the structure of the texture, the structure of the nematic order on the surface of Hydra. And now, of course, there's a lot of missing here. We still eventually, this is really the very last stage, that Hydra regeneration. In the early stages, you have a lot of plus or minus 1F on the surface, which are highly motile and eventually recombine, leading to this structure. So the question I would like to ask then is, how do defects move and reorganize in such an active nematic solid? How is this different from what happens in the liquid? To do this, I need make the model a little bit more complicated and actually include some activity. And I will model what I call essentially a nematic elastomer that is described in terms, again, of my order parameter Q, but now instead of flow, I couple to elastic deformation through a strain that I will call epsilon. So at the level of the free energy, the nematic will be again described by a Landau-Dejean free energy, which contains the double well potential. So for A positive, you get an isotropic state. For A negative, you get a nematic state. And the nematic elasticity. The elasticity of the system is just very conventional two-dimensional elasticity in terms of a shear modulus and the compressional modulus and quadratic in the strain epsilon. When I couple the two, there are a couple of terms that come about that are important. First of all, there is this term that says that the nematic tends to align with the strain. This is actually well known in nematic elastomer. It leads to what's called the soft elasticity of nematic elastomer. And it simply says that if you apply a deviatoric strain to your system, it does tend to align with the principal axis of your strain. The other term circled here in green is actually a little bit less familiar. It's what we call strain ordering. The trace of the strain is essentially a change in density. And density increase is one of the things that can drive nematic order. So what this term says is that a local isotropic strain can actually either order or disorder the material locally, depending on the sign of this coupling constants chi. So in particular, if chi is positive, chi b is positive, a negative isotropic strain, which corresponds to a compression, will actually order the system. And the dilation will actually lead to local melting. In other words, even if this A was positive and you initially are in the isotropic state, if the system gets compressed and these filaments kind of get brought closer to each other, you can actually get local order. In the opposite case, the dilation can actually lead to melting, to local melting. Now, this is still just a free energy. So in principle, these terms are all allowed in an equilibrium system. So what's the difference about introducing activity, which in this case is mostly contractile activity due to these actin fibers. Now, when I introduce activity, what I'm going to be interested again is the relaxation. Well, the relaxation of the nematic will be controlled by gradients of the free energy. And the relaxation of the strain or the elastic deformation, I will assume it's just controlled by force balance. This is equivalent to saying that stresses relax very quickly while the nematic relaxation follows more slowly, because I'm neglecting time dependence in the elasticity equation. the stress itself will have an elastic component given by the derivative of the free energy I showed you before, and an active component. And now I have both deviatoric active stresses due to these force dipoles I mentioned earlier, which have the same form essentially as you have in a fluid. But in addition, activity can also contribute to sort of an active pressure that can drive this local ordering or disordering effect together with my coupling Ib. So in some sense, activity here doesn't really change the structure of the stress tensor, but allows you to control these elastonematic couplings and possibly even to change their sign. And then I will look at the couple dynamics of this system and ask about the steady states. So what I have here, what these equations incorporate, is essentially the fact that nematic order can drive active stresses, which in turn can drive strain. And there is a feedback between the two through this strain alignment and strain ordering effect. Kind of like in a fluid, I had a feedback between flow and nematic order. Here I have a feedback between elasticity and nematic order. So here I'm not incorporating the role of the morphogen. First, in the first part, I talked about morphogen and nematic texture. Now here I'm coupling nematic texture to elasticity. And eventually, of course, I would like to put them all together. So what can I do with this little bit more complicated model? Well, let's look at a very simple realization of it. So what I can do is the force balance equation now says that active stresses can act as a source of strain. And in particular, I'm asking how active stresses can act as a source of isotropic strain, that is compression or dilation. Just like in the fluid, the equation that tells me that controls this behavior is linear, and so I can actually solve it. I can put in the texture, the shape of Q corresponding to, say, a +1F defect, and calculate the strain that that texture generates. So this kind of shows pictorially that for a +1F defect, which is contractile when the alpha is positive, I will have a compression at the tail of the defect, and I will have a dilation at the head of the defect. And this is shown a little bit more quantitatively here, where I've actually calculated, I've solved the equation. Red is compression, blue is dilation. Now, in the compression, I think I mixed up my sign possibly here because, let me see. Okay, sorry. Somehow my arrows has disappeared. The compression orders, the dilation melts. Now, the defect core is in a disordered region, and it is energetically favorable for the defect to be in a region where there is no nematic order. And so this anisotrope, this gradient of strain across the defect core causes the defect to move towards the melted region, which is the disordered region, and therefore to move in the direction of this green arrow. So there is a mechanism for motion of the defect here, which is not through flux of material, but simply to remodeling of the structure due to this sort of strain melting effect. It's sort of like dominoes falling, if you like. When you have dominoes and you get them to fall in a particular patterns, nobody's moving, no pieces of dominoes are being transported, but they're just reorganizing. And you can actually calculate so strain gradients lead to motion of the defects, and you can calculate the velocity of the defects. And in this case, it's not the sign, the direction of motion. It was a video, I forgot it. The direction of motion is not driven just by the sign of activity, but is driven by the sign of both these two parameters, activity and this coefficient chi b. And it's interesting because chi B presumably is driven by biochemistry, and this allows the direction of motion to be controlled by other biological processes in the system. So we have a mechanism for defect motion here, and the same mechanism, for instance, can actually stabilize defect structures that are not allowed in equilibrium, and might explain what is seen, for instance, the merging of two +1F defects, which is seen at the foot of Hydra. Here is shown again in a 2D geometry. So if you have a disk with normal anchoring, now you require a net +1 topological charge. But the lowest energy for such net +1 topological charge, in the absence of any activity, would be one where the defects are actually at the boundary, because due to Coulomb repulsion, they like to be away from each other. But this active coupling allow the system to overcome the Coulomb repulsion and actually get a structure of a +1 defect, as it is observed experimentally in Hydra. So this is all. We've only so far done this in the plane, but we are very interested in continuing to study these sort of active elasto-nematic and put it on curved surfaces and add the interplay of topology as well as the feedback of the morphogen. So I am going to essentially conclude here and try to summarize what I've been telling you. So in active fluids, we understand how the +1F defects becomes motile. It is simply advected by the active flows it itself generates. We can then construct a theory of defects of quasi-particles and show how defects can actually act as the drivers of these chaotic flows. And ongoing work by us and others is how to control defects in order to design active flows and achieve, for instance, laminar rectified flows out of these chaotic flows. In active solids, these +1F defects can propel themselves relative to the material through remodeling of the nematic texture without any material flow. And we have also shown how geometry and morphogen gradients alone are sufficient to understand that the texture you get in Hydra on a prescribed shape. Of course, what we really would like to do in the future is sort of closing the feedback loop, in the sense that we need to add morphogen plus curvature, promote fiber organization, and actually can focus strain and stresses at the location of the defects. The strain and stresses, what is missing from our model is the feedback from the enhanced strain and stresses at the core of the defects that can actually lead to morphogen production, and which in turn is, so strain can produce morphogen can modify the texture, and in the future we plan to try to close this feedback loop and allow, of course, for the geometry to change, to try to understand the full process of Hydra regeneration. So with that, I will stop, and I hope there might be some questions. I'd like to thank you for listening, and thank you my group here at Santa Barbara for all their work on these and other related problems.
Thank you, Cristina. Thank you very much. We really like this wonderful talk. Maybe we have time, it's a bit late for us, but maybe we have time for some questions. Stefano, please. You are muted, Stefano.
I see absolutely nothing, so only my slides. So
okay, I hope
Stefano
Ruffo. I hope you hear my voice.
I understand the Stefano Ruffo, but I'm just saying.
Yes.
Okay. I would like to make a connection between the first part of the talk and the second one. So, in the fluid case, you have shown that even at very low Reynolds number, there is a tendency to proliferate defects. So, which is the mechanism that prevents defect proliferation in the case of the solid? So, in the case of Hydra.
So in the case of Hydra, I don't know, but in this active elastonematic model we have written down, we have only looked at this in the plane so far, you can actually get something that like an elastic turbulence, meaning you can actually get a state where defects seem to be produced at very high activity, and you actually have a number of defects that are present. Now, what we have looked at in that case is, again, just a system that has activity and elasticity. We have not introduced the morphogen in any way. So we have either done morphogen and nematic texture, or nematic texture and elasticity. Okay, we haven't put it all together. So you seem to get a state where these defects are produced due to the feedback between the texture and the stresses, the strains, and so on. We haven't really quantified it. Now, clearly that doesn't happen in Hydra. In Hydra, the defects annihilate. Okay. And I don't know what is it. It could have. My feeling is that it might have something to do with geometry, with the fact that this elongated body is really flat on the sides. But I don't know. I don't know. That's a very good question. I don't actually know the answer.
And thank you.
But
thank
you
you,
do
Cristina.
get some turbulent-like behavior. I hate to call it turbulent because there's no flow here, okay? But you do get some defect generation in the solid as well.
Thank you.
Are there other questions? Apparently not. So this is probably due to the late time for us. So, Cristina,
Okay.
thank you very, very much. And the talk will be available on the website for the people who were not able to come. So we are, on behalf of the society, I thank you. So, and I hope to see you soon. Thank you again, and congratulations.